Right. Good afternoon. Thanks. Thanks to the presenter and the attendees. All the presentations have been great. My name is Joel Glagaski. I'm the Health Sciences Librarian at Georgia State University in Atlanta, and it's my pleasure to introduce the next presentation. It's called Developing a Low-Cost Interactive Histological Atlas for Anatomy and Physiology. This presentation will introduce you to the use of Pressbooks for affordable OER authorship of highly interactive materials. The presenters will introduce Procreate and the use of H5P interactive elements in OER publishing. The presenters will discuss ways to engage undergraduate students in the creation of OER materials, offering unique opportunities for professional development. The session will conclude with a discussion of the impact of OER materials on student success metrics. The session is brought to you by a team of faculty members from Augusta University. Dr. Karen Wiles is a medical educator in the Department of Physiology at the Medical College of Georgia, Augusta University. Dr. Wiles earned a bachelor's degree in chemistry from Salisbury University and a PhD from Vanderbilt University. She has taught anatomy and physiology at the undergraduate and graduate levels since 2016. Dr. Juan Manuel Ramiro Diaz earned his master's and PhD in basic biomedical sciences physiology from Universidad Autónoma de San Luis Potosí. He completed his postdoctoral fellowships at the University of New Mexico, University of Iowa, and Augusta University. Since 2020, he has served as a lecturer in the Department of Biological Sciences at Augusta University, where he teaches Anatomy and Physiology 1 and 2 and Molecular Biology Laboratory. Dr. Christina Wilson is an assistant professor at Augusta University. She has taught courses in topics including general biology, anatomy, physiology, genetics, and neurobiology. Both Dr. Wilson's graduate and postgraduate research focused on the development of an animal model of schizophrenia. Dr. Georgios Kalifatithis is an assistant professor at the Department of Biological Sciences at Augusta University. He earned his MS in biology from Darmstadt University of Technology and his PhD in cancer research from Ruperto Carola University and the German Cancer Research Center. He taught courses in anatomy, physiology, molecular biology, and introductory biology. And Soma Mukhopadhyay is an assistant professor at Augusta University College of Science and Mathematics, where she teaches physiology, anatomy, biology, genetics, evolution, among other topics. She is a physiologist by training and earned her PhD in nuclear medicine from the Indian Institute of Chemical Biology. Welcome to all of you. I look forward to your presentation. There will be a Q&A at the end, so if the audience members have any questions, please put them in the chat. And now I'm going to turn it over to the presenters.
Thank you very much for the introduction. I hope that you can hear us just fine. If you cannot, please let me know. I am super honored to be presenting today. Unfortunately, Dr. Wilson is under the weather today and was unable to join us for this presentation. We will be presenting the work that we've done to develop two low-cost interactive histology atlases for Human Anatomy and Physiology 1 and 2, which is a two-course sequence frequently taken as prerequisite by undergraduate students who are looking to enter allied health programs at the undergraduate level. So nursing, respiratory therapy, all of those types of disciplines. So the work that we're going to be talking about today is the development of two atlases and some of the SOTL work that we've done to look into the efficacy of those texts. These are the links to our textbooks. The QR codes will take you to the online OER resources for Anatomy 1 and Anatomy 2. We hope that you will share these resources with members, teaching faculty at your institutions and or librarians so that we can help them reach the widest audience that they can. These links will be at the end of our presentation as well. So the purpose of this project, and I'm presenting on behalf of Dr. Wilson, who was unable to join us, but the purpose of this project was multifold. First, we knew that we wanted to help reduce textbook costs for students. We know that textbook costs are an enormous burden to access for a lot of students, and this course is no different from many others. So anatomy and physiology at the undergraduate level is typically a 2-course sequence, and the textbook for this course is exceptionally expensive. So the laboratory manual costs are typically around $100, and the lecture textbooks are about $170. So for a 2-course sequence, that's not a bad value, since that book will see you through 2 courses, but it still can be a really large obstacle for students to have access to course materials that are of high value. One of the downsides to these commercially available texts, especially the lab manuals, is that they often contain unrelated information. So content that does not align well with commonly adopted learning objectives. And a lot of anatomy and physiology courses use learning objectives that have been developed by the Human Anatomy and Physiology Society, which is a society that a lot of the academic faculty are members of and help to kind of craft these learning objectives. So the textbooks contain extra stuff that the students don't know what to do with. They don't know whether they need to know it, and that can actually create an obstacle to success. The other thing that makes perhaps the lab manual a little bit redundant is the replication of a lot of images in both the lab manual and in the lecture text. So the picture on the right-hand side and left-hand side of this image are images from the Marab Human Anatomy and Physiology lecture textbook, which is on the right, and the lab manual on the left. And you can see that the same image appears in both. So we felt like we could maybe reduce the need for the lab manual and use the lecture textbook for a lot of that gross anatomy. One of the things that we felt that the commercially available lab manuals did not do well in terms of helping our students with really usable resources was in the field of histology. So microscope study of tissues and organs at the cellular tissue level is really difficult for our students. We know that that's an area of their coursework that's really, really difficult for them. And part of that reason that they struggle with it is that what they're seeing in their book does not align with what they're seeing on the microscope side in the classroom. So what we have here is a picture that's from the Human Anatomy and Physiology Lab Manual by Marab et al. And we see a neuron in the brain in incredibly high resolution. And unfortunately, that does not align with what the slides actually look like that the student is using in the classroom, which cannot accomplish the super high resolution and level of magnification that we see in some of the textbook pictures. We have another probably 20-fold level of magnification in this image that the microscopes that the students are using are just not capable of achieving. So this is really a huge impact on student success. If what they're looking at in the book does not match what they're seeing in the classroom, that can really impact their ability to succeed on exams. So to solve this problem, we were hoping to create a resource that was either low cost or free to the students. We wanted to be able to provide an instructional resource that was directly relevant to the learning objectives that we set forth for their learning, so that it eliminated confusion for students and made it very clear what the expectations were, so that this can help improve success. We wanted to add histological micrograph images that align with the commercially available histology slides. And throughout this entire project, the slides that we used were not textbook perfect slides like you might see in a textbook. We chose slides that the students are actually using, slides that are commercially available to all institutions from companies like Ward's and Carolina Biological Supply, so that these will hopefully be relevant to students across the state of Georgia at all USG institutions and across the country and even the globe, because these are really common representations of these different types of tissue. We wanted to make the learning interactive. We wanted to make the textbook as interactive as we could, because we knew that that would increase student engagement with the resources. And if we increased their engagement with the resources, we knew that we could increase their success academically. we wanted to finally ensure that the educational value of the resources that we produced was comparable, if not better, compared to commercially available lab manuals. And what we're comparing, the data that you'll see at the end of our presentation, is to that MERIB undergrad anatomy and physiology lab manual, which is a really commonly adopted text. One of the things that we were committed to for this project was getting students involved. We knew that if we got students involved, we could increase the pedagogical value of the resource that we were creating. But we also had a unique opportunity to be able to provide students at our institution an opportunity to get involved with this project, which would help support their professional growth and ultimately their career success. So you can see two of the images that we have here were created by two undergraduate students here at Augusta University. They have both graduated in the last 9 months, but these students, we were able to showcase their work in our textbook and allow them to graduate from college as published illustrators in an academic textbook, which is a really great thing to be able to put on your CV. Medical illustration is a field that a lot of undergrads don't really know about, but students who are interested in medicine but also have a really robust talent for art can pursue a degree in medical illustration. This is a master's level academic program, and at the current time there are only 5 institutions across North America, 4 in the United States and 1 in Canada, that offer a master's degree in medical illustration. So medical illustrators would design all of the products that you see on TV commercials and print and media surrounding things, actually the COVID, that sphere of the COVID virus that we saw so prevalently in 2020 and 2021 was actually an Augusta University graduate from our medical illustration program who works for the CDC. But there's only 5 medical illustration programs, and one of them just happens to be here at Augusta University. So we knew that we wanted to provide opportunities to aspiring medical illustrators here at our institution to help them increase their success as they pursue entrance into these medical illustration programs. So here are 3 additional examples of works that were done by undergraduate students here at Augusta University. All of these images were highlighted in our Anatomy and Physiology 1 text. We currently have 3 more undergraduate students who are working in our Anatomy and Physiology 2 atlas, and we look forward to being able to add their images to the work as it's in progress. In order to solve the problem of trying to create an open access resource, we knew that we needed a little bit of funds. We knew we needed to purchase tablets. We purchased some iPads. We needed access to illustration software. We needed to partner with some sort of digital publishing platform, and we needed some funds to be able to compensate both the faculty and the students for their hard work. And to accomplish this, we were successfully awarded 2 Affordable Learning Georgia textbook transformation grants. So a grant in round 22 for our Anatomy 1 project, and then a grant in round 25 for our AMP2 project. We're really grateful for that support from Affordable Learning Georgia. So the next part of our presentation is how did we choose the publishing platform that we went with? And a lot of self-publishing for academic texts, open access texts, is done through Manifold, which allows you to create an interactive, highly responsive, open access digital e-text. These can be hosted in digital repositories like Galileo, hosted by the USG, and in OER Commons and things like that. One of the downsides to Manifold is that it required a costly publishing partner, and it would we'd have to invest funds in layout and design and conversion to Manifold. And then any subsequent revision of the text would incur additional cost and would be quite cumbersome. So we looked for some other options around Manifold because we wanted to do as much of the editing and revision and layout as ourselves as we possibly could, so that we could keep the product as up to date as we possibly could. So we looked into a platform called Pressbooks. Publishing through Pressbooks allows you to create, again, highly interactive, responsive, open access digital e-texts. These books can be hosted in the Pressbooks directory, which is searchable online globally, and can also be linked to libraries like OER Commons and the Galileo USG library. One of the things that was really enticing about Pressbooks was that it was really affordable. The cost for the annual fee for Pressbooks is $400 per book per year. Pressbooks is committed to open access, and they only require the payment of that $400 per year in years that you're making edits. So if you do not make any edits, you do not need to pay that fee for that year, and they'll continue hosting that book at no cost as a commitment to accessibility for all users. Pressbooks uses WordPress, which is a really user-friendly interface, and it allows tech novices like myself to make really easy edits and formatting and design choices, and it works very similarly, I think, to Microsoft products. Any revisions that we make can be made in real time. We can fix a grammatical error, we can add a new image, we can delete, shift, change the layout of the book, and publish instantaneous changes at no additional cost. One of the desirable aspects of Pressbooks was their commitment to accessibility. Pressbooks is committed to being screen reader compatible, which was a really important characteristic. We wanted to make sure that our texts were accessible to all users. Pressbooks is committed to ensuring that all books and their platform in general meet the Web Content Accessibility Guidelines success criteria at either a single A or double A level, which was really important. The Pressbooks provides a user guide with a lot of guidelines for ensuring accessibility as you're creating a text, which is really important, and we utilize that guide in our design process. May make it really easy to provide alternative texts for images and caption formatting for images so that screen reader users will get a description of that image. Unfortunately, our text requires that students can see that image for maximal learning, but we did our best to try to provide descriptive alternative texts that can help students when they're using a screen reader. Pressbooks allows the text to be seamless interactivity, I guess, across all devices and screen sizes. So on the screen right now, you can see what our book looks like with a Samsung Galaxy tablet, with a Windows PC using Chrome as a browser, and then with an iPhone. So no matter what type of device students are using to access their learning resources, they're able to interact with our book with any device that they're using for their learning, without losing any of the interactive features and usability of our texts. Some of the features that we really liked in Pressbooks was the ability of students to easily navigate between chapters. So at the bottom of the screen, there are navigation tabs that allow students to navigate to previous or future chapters of the book. There's navigation to the top of the page. We also have the ability to add tables. And while students don't love tables, I think they often skip them. Tables are a really great way to simplify and organize content, particularly content that students are expected to learn specific pieces of information for and to be able to compare to other types of content. So this allows us to really just kind of condense just what we expect them to be able to know and provide on an exam. We were also able to create a glossary of terms, which is kind of a clickable pop-out glossary. There's also a chapter at the end of the book that lists all of the glossary terms and their definitions. But this allows us to define some terms that students might not already be familiar with and improve their ability to understand the material that they're reading. One of the things that we really liked about Pressbooks was the extensive use of H5P interactive elements that are available to you as publishers in Pressbooks. And one of the ones that we took advantage of was the use of interactive quizzes. So there's interactive quizzes in our Anatomy and Physiology 1 text that allow students to kind of simulate what they would be expected to do on a lab practical exam. They'll be expected to identify a tissue, so the student would type in their answer. They can get a hint for a little bit of description, something that they should be thinking about when they're looking at this tissue, and then they can check their answer and it'll provide them with some feedback about what the correct answer is and what they should be seeing when they're looking at this image. This will help them prepare for lab practical exams, which are traditionally very difficult for students. So why are we illustrating? We talked about the student illustrators. One of the things that we find is that students need a little help to imagine what they're seeing, right? When they look at a tissue under a microscope, they see a bunch of blobs that are pink and purple and a little bit blue, and they're looking really at shapes. And we as faculty can tell them what we see, but we can't show them what we see. So these illustrations help us to show the student what they should be seeing. So we have a really talented group of faculty that you'll get to meet in a minute. The rest of the faculty that are going to be speaking today were illustrators on this project, and they created these illustration overlays, which really help the students supplement a little bit of that imagination, show the students where one cell stops and where the next cell begins. What are the structures that they should be seeing on these microscope slides, and help to point out the really important pieces. So we can do this not only at the tissue level, like you see on the screen right now, but we can also do that at the organ level. So here are 3 microscope slides, one from the urinary bladder on the left, the ovary in the center, and the stomach on the right. And students might see this as a bunch of purple and pink dots, and they cannot make heads or tails of what exactly they're supposed to be seeing. But with our illustration overlays, we're able to not only label the structures, but also use color in these illustrations to help students make heads or tails of what are the important structures that we expect them to know. And with that, I'd like to introduce one of our talented illustrators, Dr. Georgios Kalafatidis, which he will talk to you about Procreate, which is our design software.
So how were the illustrations created? We use Procreate, which is a very powerful illustration program. It was originally designed for iPad Pro. There's also a version for the iPhone. And you can use your fingers to draw, but it's also compatible with Apple Pencil, which makes it much easier to make your illustrations. Similar to other illustration programs, it utilizes like layers. So you can stack your image elements one on top of the other. It has like 120 hertz refresh rate. This allows for a very smooth motion and also a very high responsiveness. And it utilizes Valkyrie graphics engine. And this allows you to use like over 200 different brushes. You can also import brushes from other illustration programs like Photoshop. And there's a brush studio where you can customize and edit your brushes. You can optimize them. So if you compare the Procreate with a very popular illustration program, Adobe Illustrator, and the pros and the cons. So Procreate relies on raster-based graphics. So it uses pixels to store the graphic information. So Procreate would be the better program for sketching. On the other hand, Adobe Illustrator uses vector-based graphics. So it uses those graphics, they use mathematical equations to precisely place a point on the document. And Adobe Illustrator would be the better program if you want to scale your work. So now what are the advantages of Procreate? So first, it has a very user-friendly interface. So there are only 2 rows of tools on the top left and top right of your screen. And on the left side, there is a slider where you can adjust the size of the brushes and also the opacity. So on the top right, you can see like one of the tools, it allows you to choose like your brushes. And as I mentioned before, there are 200 different brushes that you can use. And there's also a much broader range of colors and shades that you can use in Procreate compared to other illustration programs. And last but not least, there are also some other features that help you to speed up the workflow. So you can use your fingers to rotate the canvas, to zoom in and zoom out. You can use the 2-finger tap to undo like the brush strokes and a 3-finger tap to redo your brush strokes. And last but not least, you have another feature, the time-lapse recording feature. So every single brush stroke is actually recorded, so you can go back and watch the recordings and analyze your workflow and improve it. And also at the same time, it will let you know like how many hours you actually spent on your illustrations. So taken together, like Procreate is a very intuitive software. It has a very soft learning curve. I had no experience with illustrations programs, so it was very easy for me to learn. And also, in addition, there are a lot of tools like online videos that help you to learn more skills. And last but not least, the Procreate is a much cheaper option compared to Adobe Illustrator. So Procreate is a one-time subscription of about $13. On the other hand, Adobe Illustrator relies on the subscription model, which is like $10 a month approximately. However, there are also some limitations for Procreate. As I mentioned before, it relies on pixel-based graphics. The information is stored in the pixels. So when you scale your artwork, the pixels will get bigger and you will lose resolution. So that would be like a disadvantage. Another disadvantage is that there is a limit in the layers that you can use. With other programs like Illustrator, you might be able to use like more layers. And last but not least, there is no desktop version for Procreate. And now I would like to introduce another talented illustrator. He will explain more about how illustrations were created. Juan Ramiro Diaz.
Hello there. Okay, so as Dr. Karen Wiles mentioned, the main goal of these projects was to help the students. We want to create a book tool that we can use in our laboratories on a daily basis to help the students understand better histology. It is very simple for us with years of experience of checking things under the microscope to identify things when at first glance we see them, like in a specific cell, in a specific organ, in a specific tissue, and so on. But as she mentioned, for the students, that's very, very puzzling. They just basically see different tones of pink, shadows of cells in there, but they are not easy to identify. So that was the main goal for us in here. Help them to see on a slide what we actually see. And for that purpose is that we create these images. To give you an idea, this image in here is a slide of ileum, okay? Part of the small intestine. And let's say that we want to teach our students what is this specific structure in here. For me, it's very easy to identify that there is something in here that is different to the rest of the slide. However, for the student, they are not able to identify. This is actually a pear patch in there that it helps to the immunology of the small intestine. So what if we do something like this and we create an image that highlight that specific part that we want the students to see? And that's exactly what we did. We started, as Procreate allows it very easily, by creating different layers. We started by using the slides that they use in the lab in regular basis. We take images of them, and then we make first a collage, a lower magnification, what they see when they first sit on the microscope, and put it in focus, the slide at 4x. And together, we put a larger magnification so they know that this big piece in here, this 40x image, is part of this small one. So let's say that we want to let them know that there is something here that is important for them to notice. Well, the next step is then create a drawing where we underline, where we conturn all the cells, all the structures that are important for them to see. And then we color them and give them contrast. Now, a difference on this one that probably they can see that this part here in the middle is lighter. Now they can see that this part here is different to this part here. And even between the upper layers up here, we have sub-layers that we can identify. So what is the next? We need to give them names, right? So we create a bunch of labels and we tell them this part here is the stratum lucidum, this part here is the stratum corneum, and this slide is skin. So at the end of the process, we put together all the layers and we end up with one image like this that is the original, what they see on the microscope, and this one here, where we show what we want them to see. And then, as Dr. Karen Wiles mentioned too, we go to Procreate, and in Procreate, we can put them together. And that very important, very, very useful tool that is making H5P images. And using this image, we can juxtapose those images. We can put them on top of each other, and we can make a game. We can make the students go there and slide and hide or show or drawing in top of the original slide. So they can know now, okay, so this is what I'm supposed to see, and this is what I see in the original image. And now I know also what is the name of that specific layer. To be honest, this is super engaging and plain fun to do. You go back and forth with the slider, and you learn as you do it. On top of that, it's also useful, not only because it's engaging, but also because the students can use it to quiz themselves. They can say, okay, well, I learned right now that this layer here is the stratum granulosum, and let's see if it's true, and reveal it. And then, well, we did this for all images that we use in Anatomy and Physiology 1. And was that the acceptance for our book that then we decided to move forward and make an Anatomy and Physiology 2 book too? So actually, for this Anatomy and Physiology 2 was even more useful, because in Anatomy and Physiology 2, we review more organs, and organs are way more complex. So we can also divide in the layers, divide and say, okay, this part of the organ is a gland, this part of the organ is a smooth muscle. We have different layers of a smooth muscle in this organ, and so on. And at this point, well, your imagination is the limit, right? If you have an organ that have parts that express different types of tissues, or they have different types of glands, then you can put them, and you can make more complex collages like this one here that shows trachea. And if you see in the upper image here in this region, we have a serum mucus gland that is actually not visible in this other region of the same slide. So you can play and you can add more things. And actually, this is a continuous process of improvement that we have been going through. At that point, that now we also are adding macroscopic structures. Like this one in here, for example, that is an illustration of the heart, where we list all the structures, all the arteries, veins, etc., chambers that we are interested for the student to see. And we can even go even further by combining both types of a structure, macroscopic and microscopic, which is actually very important for organs like this one, the kidney, where we describe the structure of the nephron in this illustration in here, where we can see the vasculature associated to the nephron, and we also can see how this nephron fits in the kidney itself in the macroscopic structure. And this being said, I will hand the controller to Dr. Soma Mukhopadhyay, who's going to explain the outcomes of our projects. Thank you.
Good afternoon again. So as you have seen, our team members have presented to you starting from the purpose, our intent of developing these atlases. The reason we separated those into two parts, because the course Anatomy and Physiology are being offered in 2 sequence courses in our university, and most of the institutions nationwide as well. So we did some survey and then on the project, and then what will be the platform. We developed it, drawings or so. Now what? We have to see whether it is effective or not. So we designed 2 sets of questions which will be used across the department every faculty who are teaching anatomy and physiology, and then we will be collecting the data. So 11 questions were being used in Lab Practicum 1 and 9 questions for Lab Practicum 2. Mostly for anatomy and physiology 1, what happens, that's the very beginning of the course. And as you can see, and those who teach these courses, you know, these are the basic courses, but these are the important courses for those students who will be heading up to the advancement in their career associated with the medical field or the allied health science field. So we have to make sure that they understand the structures and also they master those and successfully complete the courses. So considering that, we try to select the questions from the different area. And if you look carefully, you will see that there are starting from the very basic cellular level of courses, structures. Gradually, we went up to the tissue, the layers, the organs, because this scaffolding concept is another very important thing for students to understand, which most of the time they do not have that expertise. As you have seen, Dr. Juan has explained how gradually the structures have been revealed. We tried to make those clear for the students. So what we did after we used those questions, we collected the answers, and these are the anonymously we tabulated the results, and this study is being approved by our Augusta University IRB. So percent of students answered correctly for each questions over the total response were tabulated. And then we did some comparison. First, the data were collected from students who use the publisher's lab manual, and you know that Dr. Wiles have already explained that process. And then the comparison was between the students using these interactive histology atlas. Now, I'd like to mention that the data, what we will be presenting today, is mostly on the anatomy physiology 1 lab practicums 1 and 2, because this is the first semester, we just have released the Anatomy and Physiology 2 book because we were developing it from last summer. So we still didn't get opportunity to collect the data for ANP2, which we will be ultimately reporting. So let's see what happened and how did we collect those. Data collected over 2 semesters for the publisher's established lab manual, spring 23 and fall 23, and the total number of students' responses collected, 349. Then using the atlas developed by us, our team here, we collected data again for 2 semesters, spring 24 and fall 24. Total number of responses we received, 213. So this is a comprehensive scenario to present what happened for each and every questions. Instead of giving the whole total correct answers, we tried to see, because this is our great feedback, to see whether it is working, how much working, what the students' responses are, whether they are getting the concept from where we have started. So as you can see, this shows that all the questions, what we used, the 11 questions for the lab practicum 1, and the bars, the comparison, the lighter shade from the publisher's book, and the darker one for the open access atlas developed by us over here at AU or Augusta University. We need to, statistically, always the higher the number of responses we can collect will give us a much better idea what is the trend. But still, even with these relatively smaller data set, what we can see that the resource what we have developed could clearly be compared with the published resources. Because For most of those, you can see the comparison for the students, comparison between those two resources are very equal. So that means the resources what we have developed here are working well. There are few responses, it could have been better. And as we always see that there is room for expansion and improvement, that's what we are trying to do. So what about the lab practicum 2? We see here for the lab practicum 2, maybe by this time students got the concept, how to use the atlas and those interactive features. In a number of questions, we can distinctly see that a big improvement over the publishers, established publishers resources. Of course, that makes us happy, makes us more encouraging that the purpose, the reason where we have started our resources are truly working. So then, as I have mentioned, the de-identified data for all of these were collected. And also what we did, we used a qualitative survey as well, 8.8 questions on a 5-point Likert scale, whether they like it or not. And then total responses collected on 138 and 78 responded. A very distinct trend. What we can see that the Atlas was clearly helpful for the students. Atlas helped the students to increase in their success. It is very easy to access, as you have seen from our previous speakers, that we did take a lot of time to find out the ideal and the optimal platform which will be equally effective across the board, whether it is the Android platform or the iPhone or the iPad or the PC. Over everywhere in the nationwide, you know that it is always a concern, passing grade versus the those who are not successfully completing the course, ABC versus the DFN withdrawal. So what we see a trend after using these manual, that the trend has improved. For the DFW has been reduced and the success rate has been increased. So our overall performance from overall observation from the performances, what we can see that there were some distinct improvements. Some cases there were lower, so we will try to gather more data and we will see if we need to make any adjustment or those pictures or not. And the qualitative survey, as we have seen distinctly over here, that shows the students were kind of responding positively that these resources are working very well. And then for the book usage analytics, this is an interesting data we wanted to present to you. If you do the Google, you will be able to see that. Over in Google, there were 27,000 plus visitors. Total visitors since the publication date have visited 41,000. And then in the Bing, 567 visitors. Also, we can see what is happening under the Pressbook as well, 44 visitors. So that means people in a different platform, they were finding it, they were looking at it, and then they were using it. And also in a number of institutions, 16 institutions where these resources have been, we hope that they are using it because they have used those our histology resources. So what about the future plan? We are still working, as I mentioned, we are already working on the ANP2 lab manual. We released it. There will be some other graphics might be added. Some modifications, changes, depending on the survey we will be doing. The data will be collected for 3 to 5 years. And we would like to collaborate with more students to get their work published and also to get the feedback. Along with that, we are almost at the end of our presentation. We really would like to thank Georgia State University for giving us this opportunity. And at the end, I will call all of my team members to be here together. And then University of Georgia, we are very grateful because we got the Affordable Learning Georgia Initiative grants. All our students, as you can see, we got a team of students who worked with us on this project, and with their pictures are beautiful. Again, the QR codes are here if you like to access it now, as you have heard about all the processes, and we will take any questions if you have.
Thanks so much for the presentation. That's a great resource. I was curious if what your response has been among your colleagues at Augusta University. I was wondering if there's talk of creating other open educational textbooks or resources.
This is not the first open education resource that's been created by the biology department. I feel like it's the most wide-reaching. They've replaced lab manuals in the freshman majors and non-majors with an open access, creating their own open access. I don't know that it's been as widely published. Dr. Mukhopadhyay has done a rabbit atlas for the AMP 1 and 2. So we use rabbits as a model system for dissection. And she was awarded a Affordable Learning Georgia grant several years ago to create a photographic atlas of the musculature and the vasculature for the rabbit, because that's not something that is included in a lot of commercially available manuals. Usually they do fetal pigs or cats, and we use rabbits, which is becoming a more common model system.
Right, and that one, the number-wise, it might not be these many numbers what we see here, but the countries, more than 28 countries have accessed that atlas. If you want, maybe we can put the link to the chat. Those who are interested, they can check that well as well.
Oh yeah, that'd be great.
Any other question?
I will add that we listed the students that we've included in our project, and 3 of those students are actually using completing an honors thesis project. One has, Jalen Richardson has completed his honors thesis and a creative thesis, including his works for this project. And 2 other students will be doing, Gabrielle Harris and Zoe Collins will be doing their honors theses as to include their work that they're doing for this project. There was a question about a print version of the book, and that's in our long-term goals. We would have to figure out how to capture the interactivity of the interactive elements of our book, and maybe make them a little bit more side by side instead of the interactive overlay. But we've been talking about ways that we know in Pressbooks that we can create a print version. We just need to be able to capture the illustrations effectively for the students in that print version. I think it would be a tremendously long textbook. Yes, a lot of bookstores. Ours does not offer print versions. I know that some has had to partner with a textbook publisher to offer a print version of the Rabbit Atlas at a very low cost. Our bookstore does not offer kind of in-house printing.
Yeah, they do offer, but we have to work like it is not just automatic. So, and also for students to buy that, if it is printed from the bookstore, it is not easy method, because we tried with when there was the COVID time, as Dr. Wiles already have mentioned, we did another open access resource. At that time, we have to work a lot. For the Rabbit Atlas, after we developed, thank you for the question. So we have found that some students prefer the print copy. So We partnered with Macmillan. That one also I co-authored with one of our other talented faculty members. And then at that time, we added, in addition to the rabbit, we also added the other animal models, whatever we use for anatomy 1 and anatomy 2, like the sheep kidney, cow eye, all of those. There was not a very huge market for that, but some students do buy it. But as it is already available free, all of these resources, that's the primary purpose, to give them the low cost book. Most of them are happy with downloading this.
The way that we kind of increase usability in the classroom is through QR codes on the benchtop that will direct students directly to the chapter that's relevant to that week to make it a little bit easier for students to navigate directly to the content that we want them to pull up. But the content is really accessible even on something as small as a smartphone. It will automatically size the images to the device that you're using. So it'll automatically kind of customize the images and the text to the device that you're using. So even on a smartphone, the students can still interact with that material in the classroom, but it's especially helpful at home. And we know, especially through COVID, that if a student missed class 1 week of lab, they were really behind, and it was really hard to replicate that classroom experience, particularly when they needed to quarantine for COVID and they missed 2 weeks of class. They really got behind very quickly. So this allows us to kind of replicate a little bit of what they would be seeing in class so that they can reinforce that content at home.
All right, thank you. Are there any other questions?
Don't know. I cannot see the chat. Do you know if there are any other questions in the chat?
If you stop sharing, maybe the stem.
There are none in the chat
Okay.
at the moment.
Okay. I know that we're a little bit ahead of our time, and I apologize for that, for not quite taking as much of the time.
No problem.
That is totally okay. What we can do is take a short 10-minute break, and we'll be back at 3.
Thank you very much for the opportunity.
Thank you.